Hi and Assalamualaikum. My name is Nur Azra Hanis.
Assalamualaikum and hi. I am Adrianna Amani Binti Mohammad Ramdzan, and today we are going to explain about an article entitled Heptavalent O-Antigen Bioconjugate Vaccine Exhibiting Differential Functional Antibody Responses Against Diverse Klebsiella Pneumoniae Isolates. Before diving into the full details, let me first explain the abstract so you get the main idea of this research. This study is about a new vaccine for Klebsiella pneumoniae, a bacteria that causes serious infections in newborns and is becoming harder to treat due to antibiotic resistance. Since no vaccine is currently available, researchers created a bioconjugate vaccine targeting 7 common O-antigen types, which are sugars found on the surface of the bacteria. They tested the vaccine in mice, and each type produced strong immune responses. However, the antibodies mostly reacted only with the specific O-antigens they were made for, showing limited cross-reactivity, meaning the antibodies produced by the vaccine against one type of O-antigens do not strongly recognize or respond to other types of O-antigens. The antibodies could bind and kill many Klebsiella pneumoniae strains, but were less effective when the bacteria had a thick capsule which blocked access to the target. In conclusion, the vaccine shows promise but may need further improvement to protect against all Klebsiella pneumoniae strains.
Klebsiella pneumoniae is a gram-negative bacterium responsible for a serious infection like pneumonia, urinary tract infection, and neonatal sepsis. It mainly affects vulnerable groups such as newborn, elderly, and immunocompromised patients, with a rising concern that it is antibiotic resistant, which make the treatment increasingly difficult. In fact, in low and middle income countries, it is now the leading bacterial cause of death in children under 5 years old. Currently, there is no licensed vaccine against this pathogen. Two key surface structures, which are the capsule and the O-antigen of lipopolysaccharide, are considered strong vaccine targets. However, the capsule is highly diverse with more than 100 types, making it difficult to cover in a vaccine. In contrast, there are only 11 known O-antigen serogroup, and 4 of them are O1, O2, O3, and O5. And they account for over 80% of global clinical isolates. This study aimed to develop a heptavalent vaccine targeting the 7 important O-antigen subtypes using a bioconjugation technology.
There are many methods used by the researchers to conduct this study. First, the researchers made special plasmids, which are small circular DNA containing genes that produce different O-antigen types from Klebsiella pneumoniae like O1v1 and O2v2. These plasmids were inserted into E. coli or capsule deficient K pneumoniae so that the bacteria could produce the needed surface sugars known as O-antigens. Next, these sugars were linked to a protein called EPA using a bacterial enzyme. This created what we call bioconjugate, a molecule that combines sugar and protein. This form helps the immune system recognize and respond better. They made both single-type monovalent vaccines and a combined version with all types called a heptavalent vaccine. After creating the vaccines, they were purified using several steps like chromatography, which helps separate and clean the molecules. Then they were checked using 3 methods: Western blot to confirm the sugar and protein were joined, mass spectrometry to check the size and structure, and NMR, which looks closely at the sugar structure. To understand how well the vaccine works, the scientists also created mutant versions of K pneumonia. This had some genes removed, for example, capsule deficient strain, so they could see how the outer layer or capsule affects vaccine performance. The vaccines were then tested in mice. The mice were given injections on days as 0, 14, and 28, using either one O-antigen type or a mix of all 7. Blood was collected on day 42 to check their immune response. To measure how well the vaccine worked, they used an ELISA test. This checked how many antibodies the mice produced against each O-antigen. They also did a bactericidal test where they mixed the mouse blood with leaf bacteria and complement, which is a part of the immune system, to see if the antibodies could kill the bacteria. Lastly, they measured the amount of capsule on the bacteria using a test for glucuronic acid, which is a part of the capsule. They found that more capsule made it harder for the antibodies to bind, reducing how well the vaccine worked. These steps help the researchers understand how the vaccine performs and what challenges need to be addressed, especially the role of the bacterial capsule.
The researchers designed a heptavalent O-antigen bioconjugate vaccine called O7V-EPA, targeting O1V1, O1V2, O2V1, O2V2, O3, O3B, and O5. These O-antigens were genetically expressed in E. coli linked to a carrier protein, EPA, and purified to make individual monovalent vaccine components. Each monovalent vaccine was tested in mice and showed a strong IgG immune responses specific to its matching O-antigen. Some mild cross-reactivity was observed. For example, O1V2 antibodies also react with O2V1. But generally, each O-antigen needed to be included individually due to low overlap. All 7 vaccines were then combined into the O7V-EPA heptavalent formulation, and this was administered to a male and female mice, and both groups produced significant antibody responses to all 7 O-antigen. No difference were observed between sexes of male and female. Next, the team assessed bactericidal activity using serum bactericidal assay, and they found 6 strains were highly susceptible with more than 50% killing. Another 6 strains showed moderate killing, around 6 to 38%, and another 6 strains were resistant with less than 5% killing. Again, high capsule producing strains were generally the most resistant. A clear negative correlation was found between capsule amount and antibody function. And the more capsule present, the lower the killing effectiveness. Strains with hypervirulence gene also tend to resist antibody binding and killing. So, although the vaccine performed well, overall, the capsule thickness significantly reduced its effectiveness in some strains.
So what can we discuss here is the researchers developed a vaccine that targets 7 types of Klebsiella pneumoniae or antigens, which are sugars found on the surface of the bacteria. These sugars help the bacteria hide from the immune system, so they are good targets for vaccines. When tested in mice, the vaccine worked well. It produced antibodies that could recognize and kill many different bacterial strains. However, the study also found that the antibodies were very specific. That means antibodies made for one O-antigen didn't work well against others. So this vaccine needs to include many types to give broad protection. One interesting and important finding was the role of the capsule, a thick outer layer on some bacteria. The capsule made it harder for the antibodies to reach the O-antigens. As a result, the vaccine didn't work as well on those strains with thicker capsules. In my opinion, this study is a big step forward in developing a vaccine for Klebsiella pneumoniae, which is a serious and growing health problem. I really like that the researchers use a smart and targeted approach by combining 7 O-antigens in one vaccine. It shows how detailed and focused vaccine design can be. However, the challenge with the capsule is important. I believe future vaccines should find a way to either weaken the capsule or include other targets like capsule antigens, not just O-antigens. Overall, I think this study is meaningful, creative, and brings hope for better protections against one of the most dangerous antibiotic-resistant bacteria, which is Klebsiella pneumoniae.
So in conclusion, this study successfully developed and tested a heptavalent O-antigen bioconjugate vaccine targeting the most clinically relevant Klebsiella pneumoniae O-antigen subtype. It showed strong immunogenicity and bactericidal activity in mice against a wide variety of strains. However, the results also highlight a major challenge, such as the strain with the heavy capsule production able to resist antibody binding and killing even when the antibodies are effective against the O-antigen. This shows that capsule interference is a major factor limiting in a vaccine performance. Importantly, this is the first vaccine to include both a major O-antigen group and a key subtype, providing the broadest coverage reported so far for an O-antigen-based vaccine against Klebsiella pneumoniae. Overall, The findings suggest that while this vaccine is a promising step forward, future strategies may require combining O-antigens with capsule-based target to achieve a broader protection, especially against hypervirulent or highly encapsulated strains. This study offers essential insight for designing the next generation of multivalent vaccines against this increasingly resistant and dangerous pathogen. That's all from me. Thank you.